Young roots can be divided into 4 regions or zones. These regions are called the root cap, the region of self-division, the region of elongation, and the region of maturation. The root cap, which is the first region, is a distinct structure, but the other 3 regions do not have sharply defined boundaries, while each region gradually merges into the next. The root cap. The root cap is composed of a cluster of parenchymal cells covering the tip of the root. The root cap is quite large and obvious in some plants, while in others it is nearly invisible. One of its functions is to protect the delicate tissue behind it as the young root tip pushes through abrasive soil particles. Root cap cells have an average life of less than a week. If they are removed, a new root cap will be produced. Until the new root cap forms, however, the root seems to grow randomly instead of downward, suggesting that the root cap also functions in their perception of gravity. The next region is the region of cell division. Cells in the region of cell division, which is composed of an apical meristem, a tissue of actively dividing cell, in the center of the root tip, produce the surrounding root cap. Most of the cell division take place next to the root cap at the edges of this inverted cup-shaped zone, located a short distance behind the actual base of the meristem. Here, the cells divide every 12 to 36 hours, while at the base of the meristem they may divide only once in every 200 to 500 hours. Cells in this region are mostly cubical with relatively large, centrally located nuclei and a few very small vacuoles. In both root and stem, the apical meristem soon subdivides into 3 meristematic areas. The first is the protoderm, the second the ground meristem, and the third the procambium. The protoderm gives rise to an outer layer of cell, that is the epidermis. The ground meristem, to the inside of the protoderm, produces parenchymal cell of the cortex, while the procambium, which appears as a solid cylinder in the center of the root, produces primary xylem and primary phloem. So, the protoderm produces the epidermis, the ground meristem produces parenchymal cells of the cortex, and the procambium produces primary xylem and primary phloem. The pith, which is a parenchymal tissue which originates from the ground meristem, is generally present in stems but is absent in most dicot roots. Grass root, which is a monocot, and those of other monocots, however, do have pith tissue. The third region, which is the region of elongation. In this region, which merges with the apical meristem, usually extends about 1 centimeter or less from the tip of the root. As the name implies, in this region the cells become several times their original length and somewhat wider, that is, it elongates. At the same time, the tiny vacuole merge and grow until 1 or 2 large vacuoles, occupying up to 90% or more of the volume of each cell, have been formed. Only the root cap and the apical meristem are actually pushed through the soil, because no further increase in cell size takes place above the region of elongation. The usually extensive remainder of each root remain stationary for the life of the plant. If cambium is present, however, there is normally a gradual increase in girth through the addition of secondary tissues produced by the cambium. And finally, the region of maturation. Most of the cells mature or differentiate into various distinctive cell types of the primary tissue in this region, which is sometimes called the region of differentiation or root hair zone. The root hairs, which absorb water and minerals, adhere tightly to the soil particle with the aid of microscopic fiber they produce and greatly increase the total absorptive surface of the root. When a seedling or young plant is moved, many of the delicate root hair are torn off or die within seconds if exposed, greatly reducing the plant's capacity to absorb water and minerals in the solution. This is why plants should be watered, shaded, and pruned after transplanting until new root hairs have formed. The life of average root hair is usually not more than a few days, although a few may live up to 3 weeks. The cells of the cortex, a tissue composed of parenchymal cells lying between the epidermis and inner tissue, mostly stores food. This tissue, which may be many cell thick, is similar to the cortex of stem, except for the presence of an endodermis at its inner boundary. An endodermis is rare in stems, but so universal in roots that only 3 species of plants are known to lack a root endodermis. In some roots, the epidermis, cortex, and endodermis are sloughed off as their girth increases. But in those roots where the endodermis is retained, the inner wall of the endodermal cell eventually becomes thickened by the addition of alternating layer of suberin and wax. Later, cellulose and sometimes lignin are also deposited. Some endodermal cells, called passage cell, may remain thin-walled and retain their strips for a while. An important tissue, collectively referred to as the vascular cylinder, lies inside the endodermis. Most of the cells of the vascular cylinder conduct water or food in solution. But lying directly against the inner boundary of the endodermis is an important layer of parenchymal tissue known as the pericycle. This tissue is usually 1 cell wide. The cell of the pericycle may continue to divide even after they have matured. Lateral branch roots and part of the vascular cambium of dicot arise within the pericycle. In most dicot and conifer roots, the primary xylem consists of a solid central core of water-conducting cells, such as tracheids and vessels. This first root xylem usually loosely resembles the rear view of a rocket with fin. Lateral roots arise towards the outside of this xylem arm from within the pericycle. Dividing cells cause the pericycle to bulge out into the cortex, and the endodermis usually ruptures before the lateral root emerges. As the new root emerges, it pushes aside or crushes the cortex cell and eventually breaks through the epidermis. Parenchymal cells between the vascular tissue of the originating and lateral root differentiate into vascular elements and made a connection between the old and the new tissue. The primary xylem surrounds pits parenchymal cell in monocot roots and those of few dicots. The primary frame, which conducts food, forms in discrete patches between the xylem arm of both dicot and monocot. A vascular cambium develops from part of the pericycle and other parenchymal cell between the xylem arm and the phloem patches in most dicot and conifers. This cambium at first follows the star-like outline of primary xylem as it starts producing secondary phloem to the outside and secondary xylem to the inside, that is, the phloem grows outwardly while the xylem grows inwardly. Eventually, the position of the cambium gradually shifts so that instead of appearing as patches and arms, the secondary conducting tissue appear as concentric cylinder.